This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 304, recorded on February 8, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, delighted to be with you.
You're in Ann Arbor. How's it the weather there? Is it getting nice?
I am. Oh, it's balmy. Like here it is February, but instead of snow and ice, it's like 45, heading to 50 tomorrow.
Wow.
It's crazy.
Also joining us from South Carolina, Michael Schmidt.
Hello, everyone.
Charleston, South Carolina. Is it Chamber of Commerce weather?
No, it's a little cool, and it's, half gray, half sun. So it's February.
And does it ever snow there?
Occasionally. It has to be the right circumstance, but
Okay.
It's generally melted by lunch.
And but everything shuts down.
Oh, it's a crisis. There's no bread to be found, no milk to be found. I don't know what people do, but all the milk and bread go.
How about cheese? Is there Roquefort cheese to be found?
There is. We actually have good blue cheese because Clemson University, which is our land-grant school, actually makes its own blue cheese. That's delightful.
Wow. Well, that's smart because if you lose power and refrigeration, the cheese will be just fine.
Yep.
Speaking
of Roquefort cheese, we're going to talk about a paper about it today, aren't we?
We are. The title of the paper is A New Cheese Population in Penicillium roqueforti, an adaptation of the 5 populations to their ecological niche. And this is a collaboration from 2 groups, that of Titania Girard at the Laboratory of Ecology, Systemics, and Evolution at the University of Paris-Saclay, which is part of the National Center for Scientific Research, better known as CNRS in France, and Emmanuel Coton from the University Laboratory of Biodiversity and Microbial Ecology at the University of Western Brittany, and they are part of the National Research Institute for Agriculture, Food, and Environment. And they study cheese and the genetics and evolution of the microbial species that we count on to make our delicious cheeses. So let me start with some background. For more than 9,000 years, long before we had refrigeration, cheese making was developed as a practical way to store food, preserve the nutrients in milk for future consumption. And here we're going to focus on the blue cheeses. And actually the process of making cheese, I should say, is really beautifully described in a brief FAQ report from the American Academy of Microbiology called Microbes Make the Cheese. It is a pamphlet that you can download. I've linked to it on the show notes. And it describes how this beloved food is the product of living microbes who use their enzymes to ferment milk. That creates solid curds that are then aged. And during this magical ripening period, microbial activities impart the desired color, flavor, texture, and even holes like we have in Swiss cheese. So we're going to think about the biology of the microbes that are making particular types of cheese, in particular the Roqueforts. So although mass production of cheese is now common, artisanal cheeses are prized for their distinct flavors, smells, colors, textures, and they rely on a whole sequence of distinct populations of bacteria, yeasts, and molds which cooperate to generate these fine foods. But we're going to focus, as I said, on the history of Penicillium roqueforti, which is a fungus that's famous for making our blue cheese blue. And as the title implied, they're going to study 4 distinct genetic lineages. One set of Penicillium strains that were isolated from moldy silage and spoiled food. Another set that were isolated from lumber and spoiled food. And then a commercial strain that is a single clonal lineage that is used to make Gorgonzola, Danish Blue, Stilton, and other cheeses. It's a domesticated strain that's been selected for robust growth on cheese and also for generating the aromas and blue color that appeal. But also this particular strain is especially good at resisting other microbes that could come in and contaminate and spoil the cheese process. So those are referred to here as the non-Roquefort domesticated strain, and you can buy it commercially, and the big producers do. And then we're going to compare it to the more elite strain, which is called a Roquefort that is of protected designation of origin. So this is a label that you can add only if you've used just local strains. So you're not adding a commercial strain of the Penicillium. Rather, it's whatever is growing in your farm. In particular, they're also going to study a very special cheese that's only available. Only a few hundred bricks of this cheese are made by a small number of farmers in the French Alps. And it's called a Tourmagnon cheese. It's a type of Roquefort blue cheese. So again, they're not allowed to synthetically add the commercial Penicillium, but rather this essential microbe is acquired from the local farm environment. So what they do to try to understand if there are differences and how they affect the production of the cheeses, they apply a series of biological and phenotypic tests and whole genome sequence analysis and phylogenetics of 51 different strains from each of these 5 groups that I mentioned: the silage or lumber and spoiled food, the pure protected Roquefort, and also the non-Roquefort blue cheeses. And they include some of this really special Tourmagnon blue cheese. So let's first look at figure 1A. This is a phylogenetic tree that found that, in fact, these 5 different sources of the Penicillium species fall into 5 distinct families based on their genetic sequence. And that is shown in really a lovely color coding that they use throughout their paper. So it makes it really easy to associate the genetics and also the phenotypes back to the different groups.
And nice pictures of cheese also.
Yeah, and they include the pictures and also of lumber and spoiled food and silage and spoiled food.
I had some of that bread in my kitchen on Tuesday.
And what did you decide to do? Did you cut it?
It went in the trash. I didn't
Wow.
try to eat it and consume it. I was scared of it.
Wow. Well, I've heard it said that there's no such thing as spoiled food, only spoiled people.
Oh,
interesting.
David Sedaris, the comedian.
Oh, yes, the humorist from NPR.
Yep. Okay, so they took a closer look at this phylogenetic tree. It was very pleasing to see that all of the strains isolated from the lumber and spoiled food were at one arm of the tree. The silage and spoiled food isolates were in another. The 2 cheese types or the 3 different cheese types were in their own group. But if they look more closely, they noticed that the non-Roquefort cheeses, so these are kind of the commercially produced cheeses, if you look more carefully at the architecture of that tree, it's got one long major branch, and then it kind of forks into a Y at the end. And that's in contrast to, let's look at the other more elite cheese from the French Alps, the Tourmagnon cheese. If you look at that map, you see that there are a lot of, it's almost like a web, especially close to the root of the tree branches. And that pattern indicates that there's been a lot of genetic recombination. So yeast, or rather, this fungus is able to mate. There are 2 different mating types. And if you have an A1 and an A2 mating type together, they can mate, form a diploid, and then genetic recombination can shuffle the genomes and generate diversity. So it's clear that this fancy cheese that's made by only a few farmers in the French Alps has undergone a lot of genetic recombination, in contrast to the kind of commercial blue cheese strain that looks, based on the whole genome sequence, to be much more homogeneous. And then if we compare the more, let's say, natural strains that come from lumber or from spoiled food and silage, you also see more of that webbing, indicating that there's more diversity, more recombination, indicating that the strains are mating and swapping genomes and recombination, et cetera. So let's look a little more carefully at the growth of the 5 different classes of these Penicillium. And they do this with a whole series of assays. They look at growth under different conditions, varying the pH, the temperature, the carbon source, also the concentration of lactic acid and salt. And the reason that lactic acid and salt are of interest is that during fermentation, the microbes in the cheese, or the milk rather, produce lactic acid, and this is a natural inhibitor of fungal growth. So it impacts the pH, but also the ability of the fungi to grow. And then salt is added because it not only enhances the flavor, but it functions as a natural preservative, because high concentrations of salt are toxic to undesirable microbes that could come in and spoil the product. So remember, this is cheese is an ancient product, an ancient way of preserving milk before we had refrigeration.
And it's often why salt was so valuable in our early days, because it was a way to preserve food so you would have stuff to eat in the cold, dark winter.
Right. So they have a whole series of bar graphs in Figure 3. And again, they're color-coded so that you can look at how the different populations behave, the different families. And in general, what they found is that the Penicillium roqueforti obtained from silage or lumber and spoiled food grew faster on sugars typical of plants, like sucrose, xylose, pectin, whereas the strains obtained from cheeses, which naturally grow on milks and proteins, didn't grow so well on these plant sugars. So that's kind of satisfying, and it's consistent with these strains adapting over their evolution with their natural environment, their natural food sources. They also found that the roqueforti strains obtained from the kind of industrial, mass-produced blue cheeses tolerated higher concentrations of salt or lower pH, more acidic pH, and also higher concentrations of lactic acid than the 4 families. And again, this makes sense if we think about where these strains come from. They are used, there's one strain that's actually available commercially and is used by a lot of our big commercial producers of cheeses that we see in our grocery stores, in these Roquefort cheeses. And so it makes sense that they have now adapted to conditions that are really specific to the cheeses. Whereas if you get the same fungal strains out from silage out in the environment, they've got to be adaptable, and they can take advantage of a wider array of food sources. So the eureka finding here is that compared to the domesticated Penicillium strains, they're commonly added during large-scale production of these blue cheeses. This more elite Tourmagnon strain from the French Alps, which naturally acquire their strains from the farms where they're grown by just a few producers. They found that there are actually 2 mating types within the 5 strains they looked at. So that is consistent with these strains being able to continue to mate and recombine and generate diversity. They also found that the 5 strains had a lot more horizontally acquired genetic elements, again, consistent with a lot more gene sharing. And in general, across their whole chromosome, they found more genetic diversity. So that was what was obvious from the DNA sequencing. But then when they looked at all the phenotypic profiles that they generated, they also found corresponding increase in diversity of their phenotypes. So their growth patterns, their pH tolerance were kind of intermediate between the other groups and were indicative of greater diversity in general. So this was a very happy result for them, because it means that they have actually isolated a natural Penicillium roqueforti strains that retain the capacity to mate and continue to mix and match their genomes through recombination. So this opens opportunities for cheesemongers, who in France are referred to as fromagers, to develop strains that have maybe different flavor profiles, different taste profiles, and also they are less at risk of accumulating undesirable inbred mutations that will cause them to lose diversity and lose other fitness traits along the way. So that point was actually highlighted by a column in The Guardian published earlier this month that reported that another key microbe in this family that is used for making camembert cheese, which this author described as the most precious export of Normandy region of France, is losing its capacity to mate. And when they looked at the strains, they found that it was losing genetic diversity. So its homogenation through this commercialization product, domestication of the cheese, is actually weakening the species. So it's an unintended consequence of decades ago when commercial cheesemakers decided to start selecting for the particular Penicillium strain that generated a white and fluffy rind on the camembert cheese during ripening, rather than its more natural green and orange mold that the producers thought was less appealing. So the overall picture here is that our large-scale production of food does deliver food more cheaply and more consistent product to a wider variety of people. But when the foods are really dependent on these natural microbes that make the cheese, that homogenation and inbreeding basically restricts the diversity, and you end up with strains that are more vulnerable and also less variety in our food. So they're very happy that they've discovered this new population, new strain of the Penicillium roqueforti, so that they can have hopes of continuing to improve the strains and also do some of their artistry and experiment with cheeses of different flavor profiles and different flavors.
Just out of curiosity, Michele, looking at figure 1, based on the appearance of Roquefort versus the Termignon, which cheese would you buy?
Depending on what my purpose was, if I was just going to be munching it by myself, I would go with the cheaper one, right? But if it was a special occasion, I'd want the artisanal cheese. Yeah.
I mean, they don't look unattractive, but, just at first glance looking at the Roquefort, it looks more like what I would think is blue cheese.
Well, it's what we've become accustomed to because it's what's mass-produced, right?
It's what's mass-produced. And the other one looks like it was made in my backyard. And, it's really about the taste. And I think that's what we have to begin to think about. It's all about the flavor profile.
Well, and also what the consumer is expecting.
Yes.
I wanted to thank one of our listeners who recommended we focus on this topic. And this was Alan Carew, who's an MD and a TWiM listener. And he had seen the provocative piece in The Guardian where they were concerned that the beloved camembert cheese was at risk, and hoped that we would discuss this on TWiM.
Well, camembert is certainly worthy of discussion. It's delicious.
It is.
Yeah, so is Roquefort.
Yeah, Roquefort's delicious too.
They are. They're making me hungry. Yes. I also we're fortunate that there are some really great tools for describing this to the general public. In addition to we've linked in the show notes to the provocative piece in The Guardian about the camembert cheese, but also a group from the CNRS who published this work have a piece called French Cheese Under Threat that describes that similar story for the camembert cheese. But they also have a 3-minute YouTube video that summarizes kind of in cartoon form the motivation for doing the research and the risk of domestication of these microbial species or our other breeds of plants or meats that we eat in the food industry. And then that FAQ from the American Academy of Microbiology that's part of ASM really goes through beautifully how cheese is made.
Michael, you remember the paper we did a while ago on wine, and they're trying to figure out what goes into good wine? It's more complicated because there are many different species there.
Yes,
Right.
And it's absolutely fascinating. And there are very few master cheesemakers in the United States, and there's only one program, I believe, in the U.S. that trains the next generation of master cheesemakers, and that's at the University of Wisconsin at Madison.
How about that?
So this is a fascinating topic. Food microbiology is an area that is often underexplored unless you're a micro major as an undergraduate and you are fortunate enough to take food microbiology. And this falls into the category of being dairy microbiology. And it's really pretty cool, especially when you move away from cow's milk to sheep's milk to goat's milk, and you really discover the wonder of the different proteins that make up milk and how it can alter the flavor profile. And it's often using the same fungi.
Yeah.
Was it Pasteur who originally showed that microbes could make cheese?
I don't know. I'd have to look at the Microbes Make Cheese FAQ to answer that question.
I thought he was the one who said.
Well, you were president of the Academy the year that came out, Michele.
And therefore, I was very busy and I couldn't read.
You were very busy and you couldn't read it, so you have an excuse.
Yeah. But I will say about cheese, I think once you've had the pleasure of tasting food or cheeses made by artists, artisanal farmers, it's really hard to eat the stuff that's wrapped in each slice is wrapped in plastic. It's not the same at all.
No.
Well, cheese has been around for thousands of years, of course.
Yeah.
People learn to do it, but I think maybe in the era, well, Pasteur was the first to start discovering microbes and their functions, right? So he may have.
Yeah.
I know.
Remember, his patron was the French wine industry.
That's right.
To compete against the German beer industry because he wanted to increase the shelf life of wine.
That's right. Anyway, I may be wrong about his role in cheese, but we can. We'll going to look it up. Well,
Send letters, folks.
When Michael is talking about his paper, I'm going to look it up. Michael, tell us what you have today.
Okay.
Thank you, Michele.
My pleasure.
This is a really neat story that I'm going to share with you. It's an open access paper that's available at MBio. It was in the January issue, and we'll put a link to it in the show notes. And its title is Yersinia bactin is a quorum-sensing autoinducer and siderophore in uropathogenic E. coli. And it's by Heffernan, Wildenhall, Tran, Kantbuta, McCoy, and Henderson, and they're from Petra's institution, Washington University School of Medicine, and they're in the Department of Internal Medicine, the Division of Infectious Diseases, the Division of Dermatology, and the Center for Women's Infectious Disease Research. So it's too bad that Petra's not with us today because she could tell us funny stories about some of these people, I think. But we discussed siderophores on and off throughout our TWiM term, but it's hard to remember which episode. But when I Googled TWiM and siderophores, I came up with episode 188, and it's entitled Turducken Antibiotics. So if you're interested, you can take a look at it. And there they combine siderophore with an antimicrobial to achieve control of microbial communities. So if you're interested. So let's start our story by explaining what siderophores are. First, and perhaps foremost, they are a public goods molecule. That concept is unfamiliar to you. Consider that the term is not of biological origin, but rather it was co-opted from the field of economics. So as you might imagine, a public good is a product or service that serves everyone. That might be a public park, the interstate highway system, or even the local sewage treatment plant or waterworks.
Or a vaccine.
How about TWiM?
TWiM. Well, the United States doesn't send us taxes. I mean, you have to go out and grovel for it. So,
But
No,
Michelle's-
vaccines.
vaccines, yeah.
vaccines would be a public good as well.
Yeah.
Their use is freely available to all. And sometimes we have to pay for vaccines, with the exception being the COVID vaccine that was free for all. Siderophores are such a microbial public good. They are produced by various microbes who produce them and send them out into the extracellular environment with an anticipation that they will chelate an ion, typically a transition element, that upon its successful return to the cell will contribute or confer betterment to the individual that has received it. Now, that public good molecule may be from the same species or an entirely different genus, but the end result is the same. The molecule returning to the microbe is contributing to the overall fitness of that microbe. Thus, in its simplest form, a siderophore is nothing more than a communal, in this case, an iron chelator, manufactured and exported, where its intended function is simply to forage the local environment for an essential element, and in this case, it's ferric iron. Another way of looking at a public good is that the microbial community is no different than our communities, in that paying taxes for something that everyone can use is considered good. The rub, of course, is no one willingly pays taxes. And in the case of microbes, production of too many public goods may serve to impact on the fitness of the organism being taxed. In other words, there's a fine line, and that takes us to the title of today's paper, that Uropathogenic, that siderophore, is actually a quorum-sensing autoinducer and functions in a way so that the microbe making the siderophore doesn't make it until there's a sufficient community that can actually benefit. And that community is stingily E. coli. Now, today's paper is a marvel in that it combines the concepts of the role of public goods in microbial community dynamics. Also, the economists / microbial ecologists call this cheating, while at the same time helping us to reimagine a role for quorum sensing beyond coordinated transcriptional control based on population density. And that quorum sensing can almost be reimagined to help us appreciate how a microbial cell has an ability to literally cheat on paying its taxes. The notion of cheating in public goods is beautifully explained in another open access paper by Parker Smith and Martin Schuster in a current biology paper from 2019. And I dropped that into the show notes. And the editors of Current Biology called this paper a primer. And apologies to Vic Dorita, who always berates me when I use the word primer, who always objects to that word. But this manuscript is just that. It's a primer. And it was appropriately labeled by the editors of Current Biology, as it serves to open our minds to the brave new microbial world of economics in the context of the nature of microbial public goods, the associated problem of cheating, which we don't have time to digress into, and ways in which microbes afford the continued production of public goods in the face of cheating, or should I say fitness pressures of living in these complex microbial communities that we are all interacting with. In our next bit of background information, quorum sensing, in its simplest form, is nothing more than coordinated transcriptional control based on population density. The process requires an autoinducer that is freely sent out of the cell, and once its concentration or density achieves literally a critical mass, it then diffuses back into the microbial cell, allosterically modifies an inactive DNA binding protein or the effector molecule, that then results in the transcriptional activation of any number of traits controlled by the now activated transcription factor. Bottom lining this thing, there's no need to make something for the good of the community unless we have a community that can benefit. And that's what quorum sensing is really all about. And if you like to see an executive summary of what the authors beautifully demonstrate and illustrate through a series of wondrous growth experiments using different isogenic siderophore biosynthetic mutants, simply jump to figure 9 in this paper. Here they present a beautiful illustration of how the Yersinia bactin is functioning as a quorum sensor induction molecule, the thing you throw over the side and say, go forage, where when cell densities are low and there is also limited iron availability, this Yersinia bactin biosynthesis is simply insufficient to match the iron uptake activity of the constitutively expressed public good iron uptake system, enterobactin. E. coli produces enterobactin constitutively. It's always starved for iron, trying to scrounge it up. And so there's no need to produce 2 if 1 is supplying the need. However, as the density of the E. coli population increases, the baseline of Yersinia bactin reaches a critical concentration in the population or the community where it can now accumulate to sufficient concentration, at which point the Yersinia bactin binds to this transcription factor, YBTA, allosterically modifying it such that now this active YBTA results in increased Yersinia bactin biosynthesis by each cell, which can then serve to bring in the much needed iron to support the expansion of the urinary tract pathogen without significantly impacting on the fitness of the community. That is to say, you don't mind paying taxes as long as your family is the one benefiting. It's really a pretty cool concept. And as Ilya would often like to say when he would condense these stories into an executive summary, that's pretty much the story. But wait, how do you know what I just offered is true? So it's now time to address the question of how did they support the description they provided in Figure 9? Again, a mini digression to set the stage and to justify how important this sort of research is. Urinary tract infections are one of the most common bacterial infections in clinics and hospitals across the globe. In the United States alone, we see over 7 million patients come in to the office for UTI and 1 million emergency room visits in the U.S. Now, you may not think that's a big deal, but the average national cost for an ER visit to treat an uncomplicated urinary tract infection is about $2,200 for patients with insurance. And God help you if you don't have insurance, it's $2,500. With the UTI being generally billed as an emergency room visit at a level 4 on a scale of 1 to 5, with the bigger number correlating with a bigger bill. 85% of UTIs result from an infection caused by E. coli, with this microbe being one of the leading causes of recurrent, that's where you get multiple urinary tract infections in successive months. No sooner that you stop taking the antimicrobial, you again become symptomatic in a few short weeks. Those are the recurrent UTIs. And many people who suffer from recurrent UTIs describe the horror show of the stress that they are continuously under. And more importantly, today, an alarming frequency of the clinical isolates of E. coli recovered from UTIs are becoming more and more resistant to our first-line antimicrobials to treat these infections. So it's no longer, here's a prescription, you go home, you'll feel better in a couple of days. In addition, they often carry resistance traits to other adaptations or fitness factors that are common to highly pathogenic variants of E. coli, where this attribute is the accumulation of multiple siderophore systems, which then can scavenge iron for nutritional use. This then serves the pathogen, as iron is most often a limiting nutrient in a pathogen's ability to become the principal microbe within a community. And the authors were right to point this out in the important section of the MBio manuscript, that while iron uptake is important for bacterial growth, the increased metabolic cost, or if you will, pain of the taxes of siderophore production could diminish bacterial fitness during the expansion phase of the infections. So what's a microbe to do? Again, these siderophore are small molecules with great affinities for iron 3, ferric, or rust, that make them competitive with the mammalian iron proteins. Remember, the tour de force in infections is lock up all the iron in human transferrin so that the bacteria can't get it to grow.
And a form of nutritional immunity.
Nutrition, exactly right.
Hide the nutrients so that the bugs can't grow.
So the binding affinity of the siderophore complexes produced by E. coli is 10 to the 52. Take that into consider. 10 to the 52. It can steal iron from anything. And this is showing you how important siderophores are to bacteria and how critical iron is for this growth-limiting nutrient. Once the intracellular iron is released into the reducing environment of the cytoplasm, the iron forms an iron sulfur complex on the ferric uptake regulator, FER, activating a feedback loop that, as you might imagine, represses further expression of the public good, enterobactin, that is being thrown out into the environment to recruit iron. No sense making more if you got enough.
Yeah, it's really a beautiful regulatory system, isn't it?
Yeah, and this is how the in silico microbiologists, the stereotypic 19-base sequence motif or the FER box, this is what they look for to see if there's siderophores out there, because it's canonically conserved. And they look in operons for genomic DNA. And I learned about siderophores at when I was a graduate student at IU. We had lab meetings with Gene Weinberg's group, and he was a great iron microbiologist. And the first time I heard the affinity constant of siderophores being 10 to the 52, I thought I had misheard. And then I immediately appreciated, and it rolls in to the tour de force of nutritional immunity and how the bacteria are fighting this battle to get the iron away from the host. Again, all this rolls into why this matters in that E. coli isolates from patients with UTI actually can encode up to 3 additional genetically conserved siderophore systems that import iron. And Yersinia bactin is one. There's samolochelin and aerobactin. One comes from Salmonella. The other comes from enterobactin. And interestingly, Yersinia bactin is a non-enterobactin siderophore that is the one that is most often recovered from clinical urinary isolates, where these strains carrying this extra trait have also been observed to carry these enhanced pathogenic potential, pathogenicity island traits that really make them superb pathogens. And actually, some UTIs can actually serve to actually kill individuals. So it's
and
nothing.
Just to put a sharper point on that, the Yersinia bactin itself is encoded on a pathogenicity island that can move horizontally.
And now this observation raises the age-old question: why E. coli strains with an elevated pathogenic potential would incur the additional cost of synthesizing additional siderophores. Now, the authors are right in pointing out there are likely two rationale that they suggest to us. The first, that these non-conserved siderophores may be more effectively able to evade the innate immune response than the Enterobacter system. That makes sense. You change the look of things and the immune system hasn't seen it before and you could go after it. And the other one that I found personally to be more attractive was these non-conserved siderophores were acting independently of iron acquisition, offering that such non-conserved ion, not iron, but ion chelators may serve to benefit infection-associated E. coli strains through interactions with non-iron transition metal ions, including my old friend copper and my other friend nickel. Recall that the life of a microbe is not easy, and that copper and nickel are both antimicrobial and often are needed for the production of essential redox balancing enzymes. And urinary tract infections are in a basically hostile environment. They're in an anaerobic environment. So redox balance, you don't have the luxury of oxygen hanging around. But we're going to get to the lab. And so it's now onto the lab. And what the authors asked of their experiments was whether Yersinia bactin was functionally redundant with the 10 to the 52 binding affinity molecule, enterobactin. It's a fair question.
Yeah. Do they really need both?
Do they really need both?
Mm-hmm.
And again, how they went about answering this question was pretty neat. First, they compared a growth model of a uropathogenic E. coli strain. And they give the number, and the number will be meaningless to you. But in addition to having the wild type, if you will, pathogen, they then have isogenic mutants that either express only the enterobactin or only express the Yersinia bactin in the presence and then absence of human transferrin. And they mix and match and they add back iron and they do have all sorts of really cool things. They chemically complement cultures of siderophore deficient strains with purified enterobactin or purified Yersinia bactin. And they go through this beautifully. And since this is an open access paper, and I cheated and provided you with the ending, I'll briefly summarize that they used a transcriptional reporter construct for siderophore biosynthetic genes to identify non-canonical modes of regulation consistent with the characteristics of quorum-sensing systems. And finally, they showed that this form of regulation, quorum-sensing, was retained in the absence of transferrin, where the community would be starved for iron, and as you might guess, would then upregulate the scavengers to go out and find the limiting resource, as well as a form of regulation that was retained when growing in human urine.
What I loved about this is that they did just some classic experiments, growth curve experiments, and they saw that the production of the Yersinia bactin was not happening so much in log phase, replicating bacteria, but rather became more abundant when they hit stationary phase. So that was their first clue that there was some regulatory difference between production of enterobactin and Yersinia bactin, and then led to this hypothesis that they tested and established that Yersinia bactin is not only able to grab iron, but it's also able to control making more of the Yersinia bactin through quorum sensing.
And they went on to offer in their discussion that this is a very attractive approach, as such a system offers the pathogen distinct advantages. First, encoding both functions with a single non-conserved mobile genetic element is genetically efficient, facilitating horizontal gene transfer. Secondly, by invoking this quorum sensing or density-dependent transcription before paying your taxes or invoking the metabolic costs of Yersinia bactin synthesis at low densities, Yersinia bactin would likely simply diffuse, offering no fitness benefit, as it would be unlikely to be retrieved by the organism paying the tax, the one manufacturing. But at higher cell densities, Yersinia bactin, sharing among producers and recycling of the now loaded Yersinia bactin, permits more rapid autoinducer accumulation while minimizing the biosynthetic costs. And as Vincent and his peers on TWiM have been preaching to the virologists, it's all about fitness, folks. It's all about fitness. And this density-dependent siderophore activity may be of particular value in that iron-limited host niches where bacterial crowding or confinement is typical.
Such as E. coli colonizing a catheter that's implanted in the bladder, so that biofilms would form, cells would be at high density, and all those cells would need a lot of iron, and so then they'd click on production of this Yersinia bactin with the amazing affinity for iron.
And then their last really cool experiment was figure 8. Yersinia bactin autoinduction in human urinary conditions. It beautifully supports the proposition that Michele just offered. In that particular experiment, what they did is they, again, have 2 strains, UT189, which is air quote wild type, and a mutant that cannot make Yersinia bactin. They're growing both strains in human urine, or the defined medium, minimal medium that they used for all of their earlier experiments, M63, which is really a bad defined growth medium. It's not very nutritionally rich. And glycerol, which is a really lousy carbon source, is the energy source. The wild type strain, or UT189, and I shouldn't really call it a wild type, but it's simpler for the audio version, is the black line in their graph. And the mutant, the one that can't make their Yersinia bactin, is blue. They then harvest the mRNA, conduct transcriptional analysis, doing your favorite technique, quantitative RT-PCR, and they show that as the population increased in size, and they verify this, they're normalizing it to a housekeeping gene, the mRNA to the Yersinia bactin became significantly greater, to the tune of 117-fold in urine. So they really want the iron from this. And in the M63 medium, it was only 9.6-fold greater than the mutant. Bottom lining this observation, the Yersinia bactin autoinduction occurred in an absence of human transferrin in both the synthetic M63 medium and the relevant human urinary environment. The last bit of their discussion is what intrigued me most about this paper, in that they offered that quorum-sensing stimulation may also optimize Yersinia bactin interactions with these non-iron transition metals, such as copper. You might ask why? Well, here's the thing. Such an interaction with copper or nickel may offer an explanation as to how the original donator of this Yersinia bactin, Yersinia, lives so well as a facultative intracellular parasite. That is, during bacterial compartmentalization in the phagolysosome, where these macrophage-like cells tried to intoxicate bacteria with copper, the synthesis of Yersinia bactin, where quorum would be achieved pretty quickly because, inside a phagolysosome is pretty small, would be especially advantageous, as Yersinia bactin would now not only have the ability to chelate iron, but also a toxic metal supplied by the phagocyte. And a further kicker, that the scavenged copper Yersinia bactin complex is known to catalyze superoxide dismutation, similar to superoxide dismutases. Now, this is a wow, because we all know how phagolysosomes kill. They kill with free radical oxygen. So now this public good molecule has potentially two functions or two benefits. First, it brings iron to the growing community, while at the same time, you have to ask, and they just throw this out at the end, but this is really food for thought. Is it Yersinia bactin functioning as a virulent determinant to defeat the phagolysosome? And if that were not enough, the copper Yersinia bactin complex has been recently described to stimulate Yersinia bactin biosynthesis, creating the next question as to whether or not Yersinia bactin system persists amongst uropathogenic E. coli due to its iron scavenging activity or its interactions with non-metal ions that are trying to kill the suckers, or both. They conclude this fine work by offering what I found to be icing on the cake. And they throw it out as the next question for all of us to ponder. What advantage does the system offer in helping us understand how host factors may exert selective pressures on these pesky pathogens that we're constantly doing battle with? And why urinary tract infections caused by E. coli are so willing to pay these extra taxes assessed on their community through the production of Yersinia bactin.
Oh, but only if they need it, right?
Only if
at high density.
I mean, this was so cool.
Let me tell you a little bit about the first author, Jim Heffernan. First of all, his most memorable moment from this project was when they tested to see if FER, the ferric uptake regulator, controlled the regulation differences between enterobactin and Yersinia bactin. He said, I was not looking forward to this experiment. The literature told us that FER was the regulator, so this experiment was to confirm what the field has been telling us. Needed to be done, but not an interesting experiment in terms of driving the project forward. However, when we knocked out FER, we discovered nothing changed in the regulation of the two siderophores, yet everything changed. I remember not even waiting until all the data were fully analyzed before running to Jeff Henderson, his PI and senior author, to tell him about it. We spent the rest of the day talking about it and brainstorming what it could mean and where to go next. The project went from looking at two siderophores that were produced differently to looking at a new regulation system for Yersinia bactin. It was definitely the turning point of the project, and it all happened from a negative result on an experiment that I was not excited for.
So important to keep an open mind, isn't it?
Well,
Controls are good.
Yeah.
just don't know where the next discovery is coming from. That's it. You have to just do everything, no matter what you feel
It's
about
a great
it.
moment. I'm happy that he shared that with us.
He wants to highlight John Wildenthal for his contribution to the paper. John was able to help with some of the experiments that the reviewers wanted us to perform. Reviewer number 3, he really stepped up and helped this paper get over the line. He's been a great sounding board to bounce ideas off, had many helpful suggestions on how to set up experiments. So John grew up in Minneapolis, and he has a special memory from when he was around 10. His mom, who's a veterinary surgeon, brought him to watch as she removed a 10-pound tumor from a Siberian tiger from the Minnesota Zoo.
And do you know how you do that? Very
Carefully.
Very carefully.
Yes.
I remember petting the anesthetized tiger and holding the tumor after its removal. This
Wow.
spurred my passion for science.
Gosh.
He went to college at the University of Wisconsin-Madison, joined a research lab because he wanted to do something he was interested in. His first lab focused on alternative fuels, ways of removing ethanol from bacteria to increase yields. Over the years, he's worked on oncology, heart disease, chronic skin, wound infections, type 1 diabetes, viral attachment to cells, viral entry into the brain, and urinary tract infections. Needless to say, I have an attraction to research and discovery of the unknown. He received his PhD in biochemistry, biophysics, and structural biology from Washington University in St. Louis, and this paper was a large part of his thesis work that you've heard about today. And he has 2 recommendations for aspiring scientists. First, don't be afraid to ask for help. I interacted with many scientists over the years in this field. I believe the most successful ones ask for help from others. You can't be an expert in everything, so why not enlist someone who is? There's always a ton of work to do, especially when it comes to trying to publish a paper or submit a grant. Use your colleagues for help. Don't be afraid to ask them if they could change your culture or run an experiment for you if you're overloaded. Sometimes, especially in grad school, people think they need to do every part of their experiments. Science is a communal effort. Use the support of your colleagues to help achieve your goals. Hey, maybe science is a public good, right?
It is indeed.
That is so smart. I totally agree. And I love to say, I can't be good at everything.
Yeah.
Well, you know, the first paper that Michele did is all about a public good because the French government is funding that work because they don't want to lose the camembert industry.
Of course.
It's big.
Well, it's part of their heritage too, right?
It's part of their heritage.
Their culture.
I mean, and you can understand, especially you don't mind paying taxes if you can eat it.
What was his other piece of advice?
His second piece is perseverance. It's huge. You're going to fail a lot. I believe 80% of my hypotheses over the research career have been wrong, and that is perfectly fine. We are studying the unknown. We're meant to stumble and keep going. Often huge discoveries come by accident or from some not thought of direction. This paper is an example of that. I discovered this production phenomenon while testing something else. I just happened to add one more strain of bacteria to a growth assay because I had extra space. Keep at it. Thank you very much.
So wise.
John.
That's another favorite expression
Jim.
of mine. It's why we call it research and not search.
That's it. Do it over and over again. Okay.
Yeah.
That is TWiM 304. You can find show notes at microbe.tv/TWiM. If you have questions or comments, please send them to twim@microbe.tv. And if you enjoy our work, we'd love your support. Financial support, microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.